The PM starts. So we are very honored to welcome Professor Laurent Franz. Laurent obtained a PhD in genomics in 2015 at Wageningen University. Since then, he's carried out research and teaching activities in the UK, at the University of Oxford, and at the Queen Mary University of London, where he is also senior lecturer in molecular informatics and genetics. Since 2020, Laurent is professor of paleogenomics at the Ludwig Maximilian University in Munich. And with his group, he exploits the power of ancient and modern genomics to contrast current patterns of genetic diversity to those in the past, tracking evolutionary processes, including artificial selection, extinction, speciation, and domestication through time. So I give the floor to you, Laurent. Thank you.
Sorry, I'm trying to unmute it. Thank you very much for the introduction. And yeah, thank you for inviting me for this. Sorry, I can't attend the whole conference because I'm already at a meeting, but I was definitely not going to pass on to speaking to you guys about what we do in the lab. So our group is at Queen Mary in London and mostly based in Munich at the Department of Veterinary Sciences. And what we are interested in is understanding the evolutionary trajectory of animals over the last few hundred years or few thousand years, particularly how humans have affected this trajectory. So we are generally interested in the impact of human activity on the evolution of animals. Particularly, one of the bigger themes in the lab is animal domestication. So how do we go from this fierce beast to this cute puppy here? And so what are the processes, sort of genomic processes leading to this transformation? We're also really interested in what happens to the genome as animals become more and more selected for specific purposes over the last 50 or 100 years. And we started working also on pathogens. So I'll show you a quick example at the end of the presentation about animal pathogens and how we can learn about their evolution using ancient DNA. And a part of my lab is also interested in understanding the impact of humans, sort of changes in land change use or hunting on wild biodiversity. So understanding modern crisis extinctions using also historical and ancient genomes. So what we do is look at ancient genomes. Why do we do this? Because with modern genomes, you really get only a picture of this long-term evolutionary processes. A modern genome only gives you really sort of a modern snapshot, and given how much of the genetics of these animals have changed over the last couple of hundred years due to heavy selections and massive changes in our biosphere, they only offer us a really sort of narrow picture of a long-term evolutionary trends. So if you want to look at what happened earlier and get a better picture of the evolutionary trajectory of these species, we really need to look back in time. And so this is why we're doing this. And so with ancient genomes, we can really start observing the evolutionary process rather than inferring with modern genomes. So our group is interested in a few broad themes. The first theme that I mentioned quickly is conservation biology. So I have a few a student and a fellow, a research fellow, Rosie, was working in Sabrina, we're working on conservation in the lab. And so what they're interested in is using molecular information from animals that they sample from museums. So they were sampled about a few hundred years ago, and the genome of modern animals, and they can compare these genomes and find areas on where genetic diversity has been going down, and they can compare this to spatial models of where land change use have happened, like here on the island of Sulawesi. So this is a project that has been ongoing for quite a few years. We're also interested in the biology of extinction. So what happens when a species goes extinct, and also as in the process of extinction. So we have a new paper on cats that I'll talk to you about, how we can use ancient genomes and historical genomes to try to understand what happens to the genome of a species as it goes extinct. our work allows us to also collaborate with archaeologists. So that means we can also address questions related to our past as a species. So we can use ancient animals to track patterns of trade in the past and patterns of human migration. So, for example, we use pigs as a proxy to understand the displaced people into Polynesia. But the biggest chunk of the work that we're doing in the lab is trying to understand animal domestication. So, where, when, and how many times species were domesticated animals specifically, and then what happened to the genome during different phases of domestication, and we also understanding the impact of industrial farming and intensive breeding on these species. So I will start with this by sort of trying to come up with a way to define what animal domestication is. So this is a picture from the 19th century and from the Museum of English Rural Life. And we can see these two kind of aristocratic looking white men that are measuring this cattle and looking at it very intensively and taking specific notes and information and sort of quantitative information about this bull here, and what they are interested in really is trying to what this message, this picture conveys, is that they are trying to use science to breed and improve these animals. And one of the things that they started doing in the 19th century is establishing breeds and pedigrees, stud books, where we starting to create populations that are reproductively isolated from each other, and that we're starting to select. And so we establishing these reproductively isolated population with specific characteristics. It's something that is really key in sort of later stages of animal domestication. But one of the big questions is, how long has this taken place? And we're thinking of domestic animals from being really from a much longer time, so cattle probably 10,000 or 8,000 years, but for how long of that process were they reproductively isolated from their wild counterparts, and were they reproductively isolated from different populations reproductively isolated from each other? And so this is really interesting to us because it's a really fundamental question in evolution, and this is a slide from a really sort of a really key review by Patrick Nussel in Trends in Ecology and Evolution in 2009, where he's trying to define the speciation continuum. So how do we get from one population or one species to two species, basically? And so one of the main important things to get two species with two different phenotypes is to maintain reproductive isolation. So he has here this speciation continuum here at the bottom here. We have weak to no divergence, the genetic level and at the phenotypic level, and as reproductive isolation increase and become stronger and stronger, we get divergence of phenotype and genotype. And so this is a process by which we think domestication is also taking place. And so how we differentiate our domestic animals from the wild, and also how we differentiate our different populations from each other, so different breeds. And so what we're really interested in one of the things that we're interested in the lab is to try to understand, how much of this is true for domestic animals in the past. We know that this is definitely true today. No breeder is going to breed their pigs with a wild boar. We don't have aurochs anymore, so we can't do this, and nobody breeds dogs with their wolf. But for how long has it been the case? How long did it take to acquire this strong reproductive isolation, and also, is it different across species? And really, we are interested in this because a lot of what we're seeing in this sort of reproductively isolated population it's really what we think is a really kind of fundamentally recent process by which people were, have applied sort of quantitative thinking the same way as we had these two old, white British men measuring this cattle. It's this idea of reproductively isolating population is fairly recent. Whereas domestication itself is thought to be a much older process. And so, for example, this is an old slide from Greg Larson, 2014. It's a bit outdated, but it's nice because it gives you a brief overview. And here we're talking about, for example, dogs. being at least 11,000 years since domestication. Actually, I'll show you in a minute. We think that this is much older than this, maybe more like 22,000 years. And here we have sheep, cattle, goats, pigs that are thought to be domesticated about 9,000 years ago. And so have they been reproductively isolated from their wild counterpart for that long? And if no, when this reproductive isolation started to take place, and is it different in different species? So I'm going to start walking you through some examples we have from walking in pigs, which is a really interesting model to address this question. So pigs were at least domesticated at least twice. We think once somewhere in China, and definitely independently domesticated also somewhere in either in the Near East, so Fertile Crescent or Eastern Anatolia, or maybe somewhere in northern Levant, in one of this area about 8,000 years ago. They were then introduced to Europe. What's interesting about this process in pigs is that as farmers were introducing pigs to Europe, they encountered a new population of wild boar that was genetically differentiated from the population of wild boar from which they were derived. So here we have a map that represents the genetic ancestry of wild boar in Europe and in the Near East, and each of the dots is a different sample. And these are modern and ancient genomes of wild boar. And so what we do is we project the ancestry of each wild boar onto the map, and that gives us this sort of 3 different colors. So we see in yellow in the Near East, so the wild boar in this region have a different ancestry from the wild boar that you find in Europe, which is represented in red. And then here we have the eastern part here in Iran, and further east, which is represented by this blue here. So what we have is, as people taking wild boar from that area here, well, domestic pigs that are derived from wild boar that should have this yellow ancestry, they're taken into Europe and they meet a different population. That's a really good setup for us to address whether was there any gene flow between these. And so we can start looking at mitochondrial DNA here, for example, which is really easy to genotype. And so here, each of these pie charts represents an archaeological site, and the size of the pie chart represents a different haplogroup. So yellow means it's something you find at Near Eastern wild boar, and red is something you find in European wild boar. And so what you can basically see is there's a lot of yellow in sort of Germany, France, and the Balkan, where actually there is no yellow wild boar. So this is really suggesting that we see the Neolithic expansion of farming, of people taking animals and plants and all these packages together to introduce farming to Europe, brought in pigs with this ancestry from the Near East. And so that's a clear signal that we can see from about 8,000 to 5,000 years ago. When you start moving closer to the present in time, however, that yellow ancestry starts disappearing. And this is the picture that we see in 5,000 to 180 years. Basically, the yellow signature has disappeared in domestic pigs and in archaeological sites generally. And so what we think is that is happening, actually, I'll just go to the next thing. What we think is happening is there's been a lot of gene flow. Mitochondrial DNA, however, is difficult to address. This question is a uniparental loci, so it's really difficult to address questions related to gene flow, because you get one or the other. It's an ancestry. So you either have the yellow or the red ancestry. You can't have a combination the two, whereas with nuclear genomes, we can definitely start looking at patterns of antrogression and gene flow. And so what we're seeing in this picture here is results of nuclear data. And so what we have here is in W is represents the genome of a wild boar, and in D is a domestic pig, and the round ones are ancient, and the square ones are modern. And what we can see is if you go back far in time in Anatolia, you get this yellow ancestry, the pure yellow ancestry, these wild boar, the modern ones, and the ancient ones. Once you start looking at sort of Western Europe, in France, for example, they clearly have this red ancestry. They have this pure red ancestry that you go back 7,000 years or today. So we have really this sort of different ancestry in these two places in the wild. In the domestic, however, as soon as you get into sort of here in Bulgaria, 7,000 years, the domestic pigs have this yellow ancestry that is very different from the local wild boar. And this is because people have brought pigs into the area at the onset of the spread of farming in this region. However, as you're moving towards sort of Britain here, and so you're moving sort of northwest, what you see is the amount of this yellow ancestry start decreasing and decreasing until it actually kind of disappears in Neolithic Britain 4,500 years ago, sort of really late Neolithic Britain. And so what this is telling us is that the fact that we find individuals like here in the site of Hexheim in Germany at 7,000 years ago that have Boss ancestry indicates that this is not a process by which people re-domesticated pigs locally, but this is a process by which pigs were interbreeding with wild boars, and they were interbreeding with wild boars to such an extent that they actually replaced the original yellow ancestry that they were coming with. And so, although this is most of the ancestry of the wild boar has been, or the original domestic pigs have been replaced, we think there is still a little bit left of that original ancestry in the genome of our domestic pigs today. And in fact, some part of the genome might actually be important and be maintained. It's sort of the original ancestry might still be maintained in some part of the genome. And one of the regions that we think this is happening in is in the MC1R gene, which is related to coat color in pigs. And so depending on whether you are wild or domestic, you have really different coat color in pigs. As a piglet, you have this stripy coat color in wild piglet, and you have this sort of yellow, so white or black coat color as a domestic pigs. And it would be really disadvantageous to have this white coat color in the wild because you would most likely be subject to higher level of predation. But here, what we can see actually is that when we're looking at ancient data, we're trying to see what the frequency of the allele coding for this white and black coat color is in the wild, for example, and for the last 10,000 years, we basically almost never see it. I think out of thousands of wild boar that we've looked at, you see it only in one individual as a heterozygote, and it's a recessive nation. So we actually don't see that phenotype and the genotype, underlying genotype in wild boar. But in domestic pig, however, this phenotype and this genotype increase in frequency dramatically and really early on. And so we see it, for example, when we're looking at science, and this is in Anatolia, and 8,300, 7,500 years ago, it's become to already 50% of the domestic pigs. The pigs found in these different sites have this high frequency, and it increased to almost about 100%, and it started decreasing a little bit in the last few hundred years. And the reason for this is the Duroc pigs. This is basically that we have pigs that have a different new allele and the red allele, and so that not 100% of the pigs today in Western Eurasia have this black or white allele, but they have a different one. But this is not the wild type. They have a different one, sort of reddish coat color that we find in the rock pigs. And we think, and this is something that we're investigating now, is to try to figure out whether this gene is in the small portion of the genome that might be still nearest in the genome of domestic pigs. So the proportions are so small that we haven't been able to really validate this yet. So we're generating more ancient genomes from all these time periods to try to map the regions of the genome now that are of nearest ancestry to try to address this question. So if we're going back to our slide from Patrick Nozzle about reproductive isolation, we can put the pigs here basically in the first part of of the slide here. So they definitely didn't have very much reproductive isolation, and they were genetically, but genetic and phenotype were still slightly divergent. So they were in between the first stage and the second stage. And so we can think of this as pigs as kind of being the norm model for what we would expect to happen if there was no selection against hybrids. So if we think about it in terms of in a population genetics context, you think about it as having these two populations of wild boars. So this population with the yellow and the red ancestry, one in the Near East and one in Europe. And then what you do is you're basically taking very few individuals from the Near East and bringing them to Europe. So the proportion of individuals in Europe that have this yellow ancestry is they are vastly outnumbered by the individuals with the red ancestry. And unless you have a very strong reproductive isolation, preventing gene flow from happening, you will basically, over time, very quickly lose that ancestry. And so what we think is happening in domestic pigs is there is just really little selection by people against hybrids of domestic and wild pigs, probably due to the way that people were keeping pigs at the time. And for the majority of the evolutionary history, pigs were sort of, almost semi-domesticated and kind of a commensal organisms that was living almost semi-managed rather than being directly managed. So really, we are here in the sort of really first part of this speciation continuum 8,000 years ago in wild boars and domestic pigs. And so really different from what we thinking about domestication today, or this view that we have domestication from the 19th century. If we're looking at dogs, though, the picture is really, really different. And so dogs are interesting because, first of all, they are the first domestic species. They were domesticated thousands of years before any other domestic species in a really different context. So people at the time were still hunting and gathering. They were living in much lower density. There was very different sort of subsistence practices that people at the time. So that's one really sort of interesting point. And so at the time when this figure was made, Gregor was thinking that domestic dogs were about 12 to 10,000 years old. In fact, our recent research suggests that this is actually probably a lot more a lot older, maybe more than 20,000 years, maybe more than 20 to 22,000 years ago. And the reason for this, I'll show you why we think this, but to put it into context, right, we're talking 22,000 years, let's say 20,000 years in dogs. It's more than 10,000 years before any other domestic species even begin their domestication process. So it's a very long term, and so domestication history, and so a lot of chances to do a lot of for interbreeding between wolves and dogs. So this is really interesting to us to look at this process back in time. And so the reason why we think that dogs were domesticated more than 20,000 years ago is because they are found in really early context in the Americas. So back in 2018 and 2021, we conducted a couple of studies where we looked at the genetics of dogs found in North America, associated with early dispersal of people. And so basically, what we make a long story short here, what we found is that the dogs that you find in North America in the past are related to the dogs that we have today in Europe, all over the world. And they came from the same population of dogs that were probably living somewhere around Siberia over 20,000 years ago. So that means that as people were going into North America and during the peopling of the America, dogs accompanied humans. And that happens at least 20,000 years ago. So that means by 20,000 years ago, there must have been domestic dogs somewhere in Siberia. Although we don't know more about where dogs were domesticated, this is as far as we go, but this provides us with at least the time frame by which dogs must have been domesticated. What's interesting about this is that we're talking, it's a very similar process of what's happening in pigs, but except for a few thousand years earlier. But when dogs are taken out of Eurasia and into North America, they meet a new wild population of wolf, a population of wolf that's genetically differentiated from the population of wolf that you find in Eurasia. And again, it provides opportunity for gene flow, and it provides us with the possibility to detect that gene flow, because the genetics of the wolf population in North America is different from the population in Eurasia. So we can ask, query these genomes and ask whether we find North American wolf in the genome of dogs that have spent thousands of years in the North American continent. And based on what we're seeing in pigs, we would expect that this would happen, because again, we would have had a vast majority of wolf compared to dogs in this area. So very few dogs were taken into North America compared to the number of wolves that were there. So if you didn't have a strong reproductive isolation, you would expect a lot of gene flow. And are really different from what we're seeing in pigs. We're seeing that actually there is almost no gene flow between these different populations, between the domestic and wild canids here. So domestic dogs have absorbed almost 0% of the genome from wolf from North America in over 20,000 years of cohabitation. And so it's very, very strong reproductive isolation here, maintained at least by people for sure to some extent in the domestic setting, but also probably maintained in the wild. We can imagine that domestic dogs at the time were already so far away from wild, from wolves, that they were not able to actually reproduce with wolves in a wild setting. They probably couldn't integrate themselves into wolf packs. But then the same thing as the other way around, that dog-wolf hybrids would not be able to integrate it in human society, maybe due to some sort of, probably due to some aggressiveness. But it's very hard to test. But what we know for sure is that they were definitely really strongly reproductively isolated. So if we go back to our slide here from Nozzle 2009, we see that dogs and wolves for about 20,000 years have been on the opposite side of the speciation continuum. They are strong divergence. They're phenotypically fairly different, although it's really hard to tell for these really early dog specimens whether they are different, but at least genetically, they're definitely different from their wolf counterparts, and they maintain these differences through a strong reproductive isolation. There is very little to no mating between dogs and wolves. In fact, all the genomes that we have looked at suggest that at least by 10,000 years, but probably more than the last 22,000 years, there's been very little gene flow between dogs and wolves. So here we're really talking about an enforcement of reproductive isolation by people in the same way that Victorians are thinking about it in a way. So I would like to move to one last species that we've been looking at. We had this paper actually that came out yesterday in Current Biology, a couple of papers that were out. So I think, yeah, this one is no more in the review debus and in press, so I haven't had time to change that slide, but a couple of papers that we've just got accepted yesterday about cats. And I think cats are really interesting, sort of counter example to wolves and pigs, about this process of reproductive isolation between wild and domestics. And so this is a result of a really large collaboration with zooarchaeologists at the University of Exeter, with ancient DNA people from my group and Gregor Larsson's group, and then from conservation genomics and population genomics just at the University of Bristol and at the Royal Society of Scotland. So cats are also interesting because we have a very similar setting as what we have seen in pigs. Domestic cats are thought to be the ancestor, sort of derived from an ancestral Near Eastern wildcat population. So here you see it in this map in this sort of yellow area here. This is a different subspecies to the European wildcat that we see in Europe here, and you see this the current day distribution of the European wildcat. So one of the big questions that we had at the time was first, validating this this link between European wildcat and domestic cats, and so addressing whether Near Eastern wildcats are indeed the ancestor of domestic cats. And using genomes, we could do this. So we sequenced the genome of quite a few Near Eastern wildcats that were sampled from areas where you find nearest and wildcat now, so the Levant mostly. And then when you compare the genome to that of other subspecies of European wildcat, but also Asian wildcat and Chinese mountain cat, you can see the nearest and domestic cat form a monophyletic cluster here, and that they derive, for domestic cats, derive most of their ancestry from nearest and wildcat. So what we are suggesting is also with zooarchaeological information is that much more recently than domestic pigs, but similarly to domestic pigs, cats were introduced more than 2,000 years ago, but not that much earlier than 2,000 years ago, into Europe after they were sort of domesticated. Well, are domestic cats really domesticated is another question, but they were introduced from a population that was genetically more similar to nearest and wildcat than European wildcat. And so that provides us with the possibility again to assess for gene flow and whether or not wild and domestic cats were reproductively isolated for this part of the evolutionary history. And again, to make a long story short, we sequenced quite a few ancient and modern genomes to try to address this question. And so the first thing that we did is to try to figure out what's the proportion of these ancient and modern genome cats, domestic cats that we have that are nearest, and what's the proportion of the genome that is European. And actually, when we're looking at this in this graph here, what you're seeing is that there's green ancestry, the European wildcat ancestry in the genome of these domestic cats. So each of these rows is a different cat's genome. And so what you can see is they have very little European wildcat ancestry. In fact, what we see here, for example, and I'm trying to find it, here in Fishbourne, so this is the oldest domestic cat that we have so far. So this is from Fishbourne, about almost 2,000 years ago, in the south of the UK, where wildcat was still around. So now wildcats are not there anymore, they've been extirpated. But when wildcat was still around, this cats found in this Roman palace in south of the UK had basically virtually almost zero of its ancestry coming from European wildcats. So it was living in parallel of wildcats that were of European wildcats with a complete different genome, which suggesting that there is little to no interbreeding between these two populations. And so this is quite interesting to us, because actually when we started looking into this in more detail, in fact, we find places in the UK, for example, where we have in the same site wild and domestic cats, so wild cats that were probably killed by people and brought back to the site in the medieval time, and animals that were also domestic. And when we look at the genome of these contemporary samples, we can find that the wildcat has no domestic ancestry and the domestic cat has no wildcat ancestry, suggesting, again, similar to what we're seeing in dogs, a very strong reproductive isolation between wild and domestic cats. And what we think is happening is basically that although the morphology of these animals haven't changed that much as a result of their association with people, their behavior has changed dramatically and their reproductive biology has changed dramatically. One of the reasons probably is that domestic cats have become more sociable. Domestic cats are able to extract resources from anthropogenic settings by able to tolerate other cats. And so they are able to tolerate other cats, which is not something that can happen in the wild. So here, and for example, in the cities like Reading, we have about 16 cats per square kilometers. In a wild cats, you would expect about 1 cat per square kilometers. And so we can imagine how this this huge difference in behavior can create a very strong reproductive barrier. A wild cat will not be able to integrate itself into a domestic environment because probably it would be too aggressive, and a domestic cat might not be able to fend for itself in the wild because he would not be aggressive enough and would not be able to maintain his territory. So here we created, without creating really a strong morphological differences, we created a really strong reproductive isolation between wild and domestic cat. And this is until we started to mess up with domestic cats and wild cats, particularly wild cats, especially what's happening in Scotland over the last 50 years, where basically wild cats have been persecuted So wildcats are being killed to a very in very large numbers in these areas, and the population are dwindling. And because the population is dwindling, it means that wildcats are making it more difficult for wildcat to find mates in the wild, and so they are starting to turn to domestic cats. And so what we're seeing is that actually you see a lot of interbreeding now happening between wild and domestic cats only in Scotland. And so here we have a principal component analysis, which each dot represents a genome, and the position of these dots on the plane here it relates to its ancestry. So here in this right side, we have all the domestic cats, and here on the left side here, we have all the wild cats. These are wild cats, for example, really old wild cat from Britain, old cats from Scotland. So all of these are wild cats. And then here you have what we call modern wild cats from Scotland. And you can see they occupy a really large part of the PCA. So number 10, for example, basically has huge amounts of domestic ancestry, whereas number 1 has very little domestic ancestry. So what we're seeing is this hybrid swarm now where wildcats are absorbing more and more European ancestry, or more and more domestic ancestry in their genome. And with ancient genomes, what we can do is really start looking at this process in time and trying to figure out when this started to happen. And so here what we have is on this side, on the right side of the y-axis, is the proportion of domestic ancestry on different genomes. So here you can see between 2000 and 2020, some genomes of wildcats have more than 75% of their ancestry that are domestic, and in some cases less than 25%, but all of them have some domestic ancestry. If we're looking at genomes from the 1980s, the 1940s, or even the 1900s, we find very little domestic ancestry here in wildcats that predates basically the 21st century. And when we model this with a program called Mosaic, try to figure out when this admixture between wild and domestic cats take place, the dates are pretty consistent to become in the really the later part of the 20th century that we've started seeing these really strong admixture. And actually, this coincides really well with this idea that this decline in wildcats. So what this suggests is basically, although we've by living next to these animals, we have established a really strong reproductive isolation between wild and domestic by basically killing the domestic population, persecuting the population in a domestic setting. We are reducing its population to an extent that we are now reversing all this long established reproductive isolation. One of the reasons why this is likely to be happening is because we think that domestic cats are not only interbreeding with wildcat, but they also bring infectious disease, and they also bring the solution to these infectious disease. So as you can see here on the right axis, when we're looking at areas of genome where of the wildcat where there is an excess of domestic ancestry, we find things like code terms like adaptive immune responses and MHC protein complex. So what we think is happening is domestic cats interbreeding with wildcat bring a disease, and then also bring the immune variability at the immunocyte to be able to actually clear this disease. So this is quite of a big problem for this population. So here we really have domestic cats and wildcat for most of the evolutionary history, at least as far as we can trace it back, are really on that really strong reproductive isolation side of the domestication spectrum, or speciation spectrum. And then we by persecuting domestic wildcat, we've switched this back all the way into the left here in Scotland, between domestic and wildcats in Scotland. So this is quite of an interesting finding that you wouldn't be able to look at without ancient DNA. So we are obviously also species can be put on this speciation continuum, but these species are probably a bit more interesting because their wild ancestors are still widespread. It's a bit more difficult to look at this in cattle or other species because the wild ancestors have gone away. In chickens, for example, there's no wild ancestors of chickens in Europe. Okay, so I'd like to move to another part of my talk before I finish. And so I have about 10 minutes. So to move to sort of different time frame and different species, and looking at a much more recent phenomenon that happens to the genome of animals that has the results of industrial farming. And so I would like to talk to you now about chickens. For chickens, we know that they were domesticated sometime in Southeast Asia, somewhere in present-day Burma, or in Thailand or south of China, somewhere around this area. And from there, they were brought into Europe, and on the way they were interbred with all these different species. And so the birds that arrived to Europe about 2,000 years ago were kind of a mosaic ancestry of different sources, but mostly coming from these Gallus pediceus. We think that looking at the archaeology of chickens, they were a relatively marginal source of food for the most of the evolutionary history of chickens. So they arrived in Europe about 2,000 to 3,000 years ago, and they stay fairly marginal at archaeological sites, especially in Northwest Europe. They really make a very small portion of the animal remains that we find at archaeological sites. And the reason for this is that they're just probably not really important. Potentially, they were simply kept as cockfighting, and maybe for a little bit of eggs, but they were definitely not raised in massive numbers. And this is up until the second part of the 20th century, where we see a huge increase in domestic chickens being raised. As many of you on this in this audience already know, we see this sort of massive rise in the production of chickens in the last 50 or 60 years. And this massive rise in the population of chickens basically created a really good ground for pathogens to evolve really rapidly. The other reason that also created a really good environment for pathogen is also the fact that we started raising these birds, which were most likely sort of free roaming backyard birds, into confined spaces like these warehouses, where infectious disease can spread really rapidly. And this is what the case of what happens most likely to Marek disease, which is a virus that affects most of the industrial poultry across the world, and that has evolved symptoms really rapidly over the last 100 to 120 years. And so it was first described in 1900, and the symptoms that it led to at the time were sort of fairly minor. It led to this transient paralysis, sort of flaccid paralysis in birds. But over time, the symptoms caused by the virus dramatically increased, and now it's incredibly virulent. One of the reasons that we think it's been evolving so quickly is because it's basically arrived at the right time in the right place. So it's a virus that has access to a huge number of hosts, a massive number of hosts. As we've seen, a lot of chickens are being grown across on the planet, and it's also extremely transmissible. So as soon as it arrived in a chicken husbandry and mass processes, we have spread of virus particle that's really quick, because they are spread via feather dust. And so they spread by feather dust, enter the lungs of the animal, and then infect them. So it spreads like wildfire in these environments. And so what it does is it creates a possibility for the virus to mutate rapidly, accumulates a lot of mutations, a lot of viruses in the population, a lot of replication, viral replication, a lot of new mutations coming in the population that provides really fertile ground for evolution. Not only do we have this, but we also have a selective pressure that applies on top of this new genetic variability that comes in the population. And this new selective pressure is potentially applied by vaccines. So the way that the vaccine works in this species is that it doesn't really prevent the symptoms, but it's sort of delay it prevents the symptom, delay the onset of the symptom, but doesn't really prevent the transmission. And so this really cool paper from Venus group in February has showed that the modeling of this has showed that if you apply these very leaky vaccines, you might apply a selective pressure onto a virus to become more and more virulent. So we have now a virus that has access to huge genetic viability and the selective pressure necessary to evolve really rapidly. The problem with understanding really what happened at the genetic level of this virus is that we don't have any information from samples prior to the 1960s. So addressing questions like for how long MDV has been circulating in the population, or what is the molecular basis for virulence, is really complicated. We have a few genomes from the 1960s and early 1970s, late 60s and early 70s, but in most cases, most of the genomes that we have are coming from strains that are extremely virulent. So we don't really know about the genetic viability that existed prior to the virulence of these species. And so this is where ancient DNA becomes really handy. We can get genomes not only from the animals that died, but also from the pathogens that were living with the animals and all sorts of other commensal microorganisms, and that were living at the time with the animals at the time of his death. And so this is something you can do for Marek disease, and we've managed to generate genomes from ancient Marek from quite a few places all across Europe. And the first thing that we've been able to show is that we think that Marek has been around in the population of chickens for at least 2,000 years. So we find it again, this is fishbone. We saw the cats from fishbone earlier. This is fishbone here. The chickens from fishbone seems to have Marek. Very low genome coverage. This is the genome coverage, but we have really good evidence that we think that Marek was circulating in the chicken population for more than 2,000 years and leaving probably little symptom or not enough symptom to actually be noticed in any veterinary treatise. What you can do with this data is you can compare the genome of the ancient and the modern strains and try to find regions where they really look really different. And this is a virus chromosome, this round chromosome, circular chromosome here. And so we can look at different summary statistics like pairwise divergence and sequences. And when you're looking at this, you find a gene called the MEK gene that's looks like it has evolved very rapidly in the modern lineages compared to the ancient lineages. So we find regions in this gene that are fixed now in modern lineages that were viable in the past. And these regions of the genome of this with the specific genes often overlap with specific amino acid changes. Often these proline repeats. So I won't have time to go into detail about this, but what we see is that we can really pinpoint in the genome and in the genes here involved in this MEK gene, which is involved in tumor proliferation, what molecular changes happened in the modern strains. The molecular changes that are now fixed in modern strains. So, for example, here the number of proline repeats in this gene used to be 6 in ancient sequences, and now it's between 4 to 5 maximum in less virulent strains, and about 3 to 2 in really highly virulent strains of Marek disease. We can then also take this sequence and do a transfection experiment, so we can sort of re-synthesize this MEK gene and do a transfection experiment and express it in the cell and look at its transactivation capabilities. And what we can see is that basically the transactivation capabilities of this ancient MEK strain is very much less, means that it's much likely, much lower transactivation capability in the ancient strain compared to the modern strains, even what they call mild strains, which are still pretty virulent strains, or compared to the very virulent strain. So what we think here is that given this low transactivation capability, the ancient of this gene, the ancient strain was unlikely to be able to cause tumor in the modern. In ancient strains were unlikely to cause tumor in the birds that it was affecting. And so this is probably the first time that we have been able to sequence a a naturally occurring strain of Marek disease that was non-virulent. And so some of the things that we're doing now is starting looking into in vivo experiments to try to validate this, not in vitro, but also in vivo. All right, I think I'm out of time, so I will leave you with this. So this is obviously a lot of collaborators and a lot of people that have been involved in a lot of these studies. And so this is our lab, and some of the major collaborators, but a lot of them are missing here. And thank you for your attention. I think there probably will be time for questions.
I hope you can hear the claps, Laura.
Yes, I can hear you.
So thank you very much for your talk. I don't know if we have any question here or online. I don't have my PC at hand. So Ricardo, do you see any question in the chat? No? Not yet? Well, so I can start. Oh, yes, Tatiana, okay. You need the microphone, please.
Hi, thank you very much for this very nice talk. I had a question concerning the dogs in America. Maybe I missed something from what you said, but from what I understood was that you didn't see any integration of wild wolves' DNA into modern dogs. Is it right?
Yes, that's correct, yeah.
And what about the vice versa? Did you find any modern dogs integration into wild wolves?
So almost none. We don't really see that much. There is a few examples, so maybe in the wolf from Yellowstone, which so in this national park in North America, which maybe acquired a few alleles from dogs that may confer them some different coat color, but that's a very rare sort of marginal occasion, and that's a very small amount of gene flow anyway. So we really don't see that much gene flow between wolf and dogs at any time in the past or today. Yeah.
Okay, thank you.
No, well, waiting for other question. Well, I will pass on cats. So
go ahead.
I find it actually surprising that cats, were introduced, you said the domestic cats introduced in the EU like 2,000 years ago, which is very recent. It seems surprising. So what is the cultural reason for this? I don't know, maybe other animals were more useful for making some works, and the dog was guarding, making guards and protecting the house, and the cat was just a parasite. Why it didn't happen earlier?
Yeah, it's a good question. Often with these things, sort of these late introduction, people blame the Romans. And so maybe this has to do with the expansion of the Roman Empire. This is what zooarchaeologists are saying. I'm not 100% sure, why cats would not be able to spread earlier. Maybe there were less rodents at sites. Maybe we didn't have dense enough sites that there was enough rodents for cats to, really be able to live. Maybe there is an adaptation that allowed them to live in colder climates. I don't know. This is a really good question. This is something we don't really know why it took so long for cats to spread to that part of the world. Yeah.
Yeah, interesting. Do you have any question? Can you check also, Ricardo, if somebody has...
There's some question online.
There is Hervé with a question. You have a question online, so no. Okay.
There was Ole had a question, I think. His hand was raised for a while. It's been raised for a while.
So meanwhile, I asked my question.
I don't know if he's reading a question.
Yeah.
No, it's fine. Okay, go, Hervé.
No, it was just whether the dog was an exception. And if you look at all the domestic species, you will see a mixture that you do not see in dogs.
I think that is an exception, yes, but the cat is quite similar. I think it really depends on how people were perceiving the animal at the time. right? So it will depend on the role that the species was playing in the society. I think in dogs, if you're talking about, roles that they were playing, maybe guarding or also sort of as pets, there probably was a strong selective pressure against hybrids. In pigs, there were just, some source of meat that was roaming around the settlement, so it was easy to, it didn't really matter whether you had a bunch of wild-born ancestry or not. So it really depends on the role the animal was playing and in the specific time frame. Yeah, does that answer your question?
Okay, thank you. Maybe we can answer, Laurent. Maybe you see the chat as well on your PC?
Yeah.
It's a
yes.
question.
There was a question from Ole that's been waiting here, so I guess.
So you want me to read it or you read it yourself? So do you see shared genes or common pathways that have been selected during the domestication of several species? Yes, okay.
We haven't really looked at this in much detail. I mean, there is a whole suggestion that there is these neural crests. There's this neural crest hypothesis that there's genes involved in migration of sort of neural cells during the development of the neural crest that might be involved. That these pathways will be involved in mass domestication, at least animal domestication. I don't know whether this is the case. I think the idea that this might be the case is not necessarily correct. I mean, I think a lot of these animals have very different biology, and the pathways to which they become domestic is very different. In some cases, there are a lot of gene flow. In some cases, there is little gene flow. In some cases, we selecting for tameness. In some cases, you don't really select for tameness. So I don't know whether we would expect to see that, but we haven't. I think people have started looking at this. It's quite difficult to really, test. I think also selection would have varied quite a lot in the past, right? So what we're thinking is like domestic animals today are really different from domestic animals that were just a few hundred years ago. I mean, pets, for example, dog pets, probably the earliest dog pets is probably a Roman thing. There was no dog pets before this. So the selective pressure that we see in these animals, in the genome of animals, probably for docility and tameness and things like this, is probably something fairly recent as well. So it's really hard to say whether sets of common genes will have been sort of selected early during the process. The sets of genes will have been selected early in the process of domestication in different species. It would be really hard to test. Yeah.
Okay, and I think Ole has raised the hand.
So. Yeah, thank you. Hey, Laurent, nice to see you.
Hi. Yeah, good to see you.
Well, I have one question about the cats. I think we have also been talking about it before. So why are there so little phenotypic differences in cats selected compared to many of the other ones, where you really see many different appearance, or much more difference in my opinion. So did you come along any suggestion for this?
No, it's a good question. I mean, I would imagine there's a selective pressure, but some sort of selective pressure to keep things the same in that morphologically. I mean, it's the most optimal form that a cat can take. And
but you didn't try to select for like a lion or something, or a lion-size cat or something to help you?
Do you mean like, did people select for large size of cats in the past, or?
No, just, I mean, we have all these different sizes in dogs, and the pigs are also quite different in size, or?
You're right, the size variability in cats is, there's very little size variability. I mean, wild cats are bigger. The European wild cats are bigger than the Near Eastern wild cat and the domestic cats. But the variability within the population is quite small. So there is not that much to select from. And in dogs, for example, we know that the variability in the population of wolves is already large. So the allele, one of the alleles that explains the largest differences in wolf or in dog sizes, so the allele that you find in Chihuahuas versus a Great Dane, these alleles were already viable in wolves. And so you select from standing genetic variation, and maybe this is not something that exists in cats. So there's nothing to select from because cats are kind of relatively homogeneous in size. So maybe this is one of the reasons. But you're right. This is a really interesting. There must be some sort of selective pressures against changing that. That's really sort of really good morphology that you want to keep. Yeah.
Well, I have one more question, maybe if it's allowed.
Yes, but we are already late.
It's a quick question,
let's
I
take
think.
2 minutes more.
So what about who is domesticating whom?
Yeah, that's a good
Did we
question.
domesticate the pig, or did the pig domesticate us?
I think, yeah, that's a good question. I mean,
Or the dog, or the wolf, or...
I think to me, the idea that definitely the species has got a lot of boost in fitness. So it's great for a species to become domesticated as a species, but as an individual, the boost in fitness is quite low, right? So most individuals in the domestic population, especially today, like we're looking at cattle, their fitness is zero, right? They're not actually being, so a few individuals have very high fitness and some individuals have zero fitness. So I think as an individual, it's not that beneficial to be domestic. But as a population, yeah, it becomes quite helpful in terms of for the species to have a high boost in fitness. So yeah, I mean, you can think of this sort of self-domestication, or at least we can think that it's beneficial for species to become domesticated. I think that's a really sort of interesting take on this. Yeah.
Thank you.
Okay,
Thank you.
so thanks again, Laurent, to be with us.
Thank you very much. Yeah.
Thank you a lot.